these works are focused on the development of a genome-scale metabolic models for different organisms. And in this webinar, we will overview some of the current activities of this team, such as the genome-scale metabolic model of the Cork Oak tree that comprises over 8,000 reactions and includes pathways of the secondary metabolism. And also we will learn about the YEASTRACT portal and we will see that some of these activities are aligned with the mission of the microbial biotechnology community at the European level. So without further delay, I will pass the floor to the speakers. And once again, thank you very much for joining us today.
Oscar, please go on. Yeah.
Can I share my screen? Okay. Can you see it?
Yes.
Okay. So thank you for this invitation, for presenting our work with the Cork Oak tree model. So this is might seem a bit strange because this is the microbiology and biotechnology community, and the Cork Oak is not a microbe, of course, and but it's a tree of national interest, and it's very important for the Portuguese economy. So regarding the reconstruction of models, as Anna said, in this community we have been developing several models for different organisms, and we have an established workflow with 4 major steps that are subdivided in smaller steps. So we start from the DNA sequence and we perform the annotation where we identify the gene products, namely the enzymes and the transporters, which are the ones that are associated with the metabolism of the organism. So we try to identify the portion of the DNA that is associated with the metabolism, with the metabolic capabilities of the organism. From here, we identify, we assign reactions to each enzyme. So one enzyme can catalyze one or several reactions, and one reaction can be associated with one enzyme or several enzymes, either complexes, protein complexes, or even the isoenzymes. Then, when we have this set of reactions, we have to identify all gene-protein reaction rules, the compartments in which these reactions take place. We might have to perform some gap-filling steps in which we try to identify which reactions are missing from a given pathway. And finally, we have to include also the transport reactions so that we can provide the model with everything it needs to be able to perform a simulation. When we gather all of this data, we can convert these networks, these sets of reactions and rules into proper models, proper genome-scale metabolic models. For this, we convert all the reaction sets into a stoichiometric matrix and a flux vector, which, following the principles of chemical engineering, we know that in steady state there is no accumulation nor degradation of metabolites. So we can say that the product of this matrix with the flux vector will be equal to 0. We have also to include other constraints like thermodynamic constraints, in which we have to determine if one reaction is reversible or not. So if the reaction can take place in just one direction or both directions. And finally, we have to include a purpose for the cell, like like formulating an equation that represents the biomass of the organism. So we usually like to create this reaction has a sum of macromolecules like DNA, RNA, protein, lipids, carbohydrates, and other cofactors. it depends also on the organism that is being modeled, the type of macromolecules that we can include, that we should include in this abstraction that represents the amount of macromolecules that we need to generate, to biosynthesize, to create another unit of biomass. And finally, on the fourth stage, we can perform the model validation by usually by performing in silico simulations in which we determine an objective. Usually for microorganisms, the objective is to maximize the biomass growth rate, subject to the restrictions, to the constraints of the stoichiometric matrix, and as well as the flux constraints. And then we compare the experimental data to the phenotype predictions, and we can sorry, there's some noise there. So, and then we can, when comparing the experimental data to the phenotype predictions, we can validate the model to see if the model is complying with how it should. So if it is behaving how it should when we compare the data. If it doesn't fit, the phenotype predictions doesn't fit the data, we go and revisit all the steps of the model to see what we missed, if it was on the genome annotation, the construction of the reaction set, or in or when we establish the flux constraints. Regarding the Cork Oak tree, as probably most of you know, it's a forest tree that is indigenous to the Mediterranean region. It produces cork, which is a valuable product, which has a huge economic impact in Portugal, though this tree is vulnerable to both biotic and abiotic stresses like climate changes, diseases, forest fires, and a genome-scale metabolic model for this tree can be very important to study how the tree adapts, for instance, to drought or heat waves, and to understanding the mechanisms that provide resistance to this stress. So the goal of our project was to reconstruct a high-quality model for Suber, a high-quality model because we want to reconstruct a manually created model. Then we wanted to integrate transcriptomics data to obtain tissue-specific models. So besides having a model that represents the whole genome, the whole metabolic potential, then we want to restrict this model to and extract tissue-specific models for leaf and for other different tissues of the tree. Then the third objective was to reconstruct a multi-model, a Diel multi-tissue model, where we integrated different tissues and different conditions to see how the tissues would communicate with themselves and perform simulations under this with this multi-tissue model to see the behavior of the multi-tissue model. And finally, we performed evaluation of the metabolic behavior of the tree under different stress conditions. So the methodology that we followed to reconstruct the model was we started with the genome sequence, which we obtained from NCBI with this accession number. This genome was submitted on December 2017 by Sebal. And initially we performed a blast against the whole genome of against Swiss prot of the whole genome, of all the ORFs that were identified in the genome. And then for the ORFs for which we couldn't find any results when blasting against Swiss prot, we blasted against Tremble, always setting any value threshold of e to the minus 30 to have confidence on the results that we obtained. Then we used all of this was performed with our tool that we developed for reconstructing models, Merlin. And then we used Merlin's workflow for performing a semi-automatic genome annotation, in which we provide a list of organisms, an ordered list of organisms in which the software should base its predictions on when annotating the function of each gene. So initially, we started with the reviewed entries of Quercus suber and Quercus gender, and then we selected other well-studied plants like Arabidopsis thaliana and Vitis vinifera and so on. Finally, for the unreviewed entries that we obtained with Tremble, we started with Arabidopsis, also because it's the most well-studied plant, and then we went for other well-studied plants, and finally, if we couldn't find any annotation, we would go with the default one. Regarding the transcriptomics data that we used for creating the multi-tissue models, we also retrieved from NCBI for the leaf and inner bark. We retrieved the data set that was deposited by Sabal, and for the phylogen, we retrieved a data set that was deposited by FCool. And after retrieving these data sets, we performed a quality check with FastQC. Then we used the SQL for the quality trimming, and then bowtie to perform the alignments against the reference genome. Then the feature counts feature from the subread package was used to perform the counting of the mapped reads. And finally, we used the HR library to normalize the read counts. And then we use this data together with the model that we previously reconstructed with Merlin. And we used another tool that we developed in-house that was developed by Vitor and George Tropo, which created our specific tissue-specific genome scale model. So together with the transcriptomics data that we obtained with the previous workflow, we integrated this data with the model, and we created specific tissue models for leaf, inner bark, and phylogen. And then finally, we integrated all these models into a Diel multi-tissue model. So it's Diel because we simulate both day and night conditions for each tissue. So for each tissue, we have two models, and then we have the common pool, which allows us to establish communication between the different tissues. So we have now 6 models, so we have 3 tissues to each tissue with a day model and a night model, and then we have a common pool of metabolites, which represents the metabolites that we are allowed the model to exchange between themselves when simulating. So regarding the results from the genome annotation, from the nearly 60,000 genes that were retrieved from the submitted genome, we identified less than 80,000 metabolic genes. Nice. Most of these metabolic genes, over 7,000, are enzymes that are transferases, followed by about over 5,000 hydrolases and over 3,000 oxyuretadases. So these are the 3 major enzyme families that are represented in this genome according to our notation. And then we have less than 1,000 for all the other 4 classes, lyases, isomerases, ligases, and translocases. So these results are already really different from their less balanced than the ones that we usually find for microorganisms. Regarding the biomass composition, so like I said, for the model, we also need to establish a biomass composition that represents the drain of macromolecules that the cell needs to produce to create a new unit of biomass. And we found that the biomass for the different tissues is also really different. So for the leaf biomass composition, we relied on the biomass composition that was established for other leaf models like Arabidopsis. The Arabidopsis model has a similar composition to this one, in which most of the protein, most of the leaf of the biomass is protein, so almost half, followed by carbohydrates, lipids, and then the cell wall, and then a small portion for DNA, RNA, cofactors, and others. So for instance, the chloroplast is included in the cofactors and vitamins pool. Then in the inner bark and phylogen, we used publications that are available where we have there's an analysis of the biomass composition, and we can see that it's really different because for the inner bark, most of the biomass are carbohydrates and the cell wall, and a smaller portion of protein. Whereas for the subring, most of the composition is for the phylogen, most of the composition is subring, and also cell wall, carbohydrates, and protein. So these are also three different, the models that we have for different tissues also have different biomass compositions besides the different set of reactions that were obtained through the transcriptomics data. So finally our model, our global model, the whole genome model is composed of 7,000 genes, over 8,000 metabolites, and 7,500 reactions, and it is comprised of 8 compartments. We have here a small sample of some of the metabolic models available for other plants. So we have the Rapid model with 5 compartments and less than 3,000 reactions. The maize model, which has 9 compartments, it has even 1 compartment more than our model, but has less reactions and less metabolites, and they don't identify genes. The tomato model has 5 compartments and less than 2,000 reactions, and the more recent rice model has a smaller set of reactions, so 1,300 reactions and 3,600 genes. On the right, you can see, hopefully, a small Venn diagram in which we compared our model with the maize model and the Raptopsis model. So you can see here that if you sum up all the reactions that compose, for instance, the Cork Oak model, you won't get the 7,500 reactions that you have on the table. That's because the reactions and metabolites are the same reaction and metabolites in different compartments are considered different reactions in this type of models. So to perform these comparisons, since the compartments are not the same in all models, what we did was we ignored the compartments and we considered the same reaction in different compartments has the one unique reaction, so that we were able to compare all, at least these 3 models. And finally, we obtained a large core set of reactions, in which for the 3 models we have almost 900 reactions that are common to the 3 models. And if we compare just Cork Oak model with the maize model, we have over 1,000 reactions that are common between these models. We can also notice that the Cork Oak model has a lot more reactions than the maize model and the Arabidopsis model, unique reactions. This model has over 1,000 unique reactions because the Cork Oak metabolism seems to be more differentiated than the other models. Regarding simulations performed with the Leaf model, so these results are for simulations when they performed with the Leaf model alone, one that we built using the transcriptomics data from CEPAL. So we calculated one parameter that is usually used to assess these models, which is the simulation quotient, which is the flux of CO2 by the O2, the oxygen that is produced in a simulation. And as expected in literature, the nitrate coefficient, the simulation coefficient is lower than the simulation coefficient when using ammonia as a nitrogen source. So this is a parameter that allows us to see how much carbon is assimilated by oxygen that is produced, and it is expected that more carbon is assimilated when using ammonia as a nitrogen source. We have here one of the Oriza Sativa model, the REST model, also has their simulation coefficient available for the different analyses that they perform, and their simulation coefficients are between 0.87 and 0.98. Another validation that is usually performed with these models is the quantum yield. which is the CO2 that is assimilated by photon that is also used assimilated during the simulation. We also calculated this quantum yield both for nitrate and ammonia, and according to literature in the autumn, this coefficient should be between 0.84 and 0.89, and somewhere between 0.51 and 0.75. We can see here that this coefficient is usually quantified through the Rubisco carboxylation oxygenation coefficient, and so normal conditions are considered when this coefficient is set at 3, and at 3 we would have about 0.07 for nitrate and 0.75 for ammonia. So these results have to be further studied because they are somewhat not as we expected. Though when we analyze here at the right the carboxylation oxygenation flux in Rubisco, we can see here that when, like I said, when at 3, which is normal, when we consider this the base flux, we can see here that when we decrease this the carboxylation oxygenation ratio, which means that the cell should be under drought conditions, so it's under stress, we can see here that the malate dehydrogenase has a behavior. So this enzyme is really, the flux decreases a lot through this enzyme, and that it affects the TCA and oxidative phosphorylation, as well as when the plant is well watered and the carboxylation oxygenation ratio is above 3, this malate dehydrogenase also increases its flux, so it has a higher fold change in flux, and it also impacts the TCA flux. Also, as expected, when we decrease the carboxylation or oxygenation ratio, there is the drought stress, so there is an increased flux in the photorespiration. So we can see here the oxygenation reaction of the Rubisco increases in flux, as well as all the reactions involved in the photorespiration pathway. And also in the Calvin cycle, we can see here that the Rubisco reaction involved in carboxylation also has a decrease in fold change, and it also impacts a little the Calvin cycle, increasing a bit the flux in the other reactions of the same cycle. So globally, we can say that results seem promising. We are complying with most of the tests usually performed with these models, and the pathway analysis of the models are showing that the model is behaving as expected when submitting the simulation, when performing simulations within stress conditions. Finally, we have the results of the Diel multi-tissue genome scale model. We show here what the simulations that we performed. So the light and the photons and CO2 were fed to the model through the Leaf light model. And since we don't have roots in this specific model, we are feeding the nitrate and the other and phosphate and sulfate through the inner bark. And we can see here that when performing simulations, only the Leaf light model is producing oxygen. All the other models are consuming oxygen, and this model is also providing the other models with sucrose and amino acids, and both went to the inner bark and to the leaf in the night simulation. When in the absence of photons, it uses the starch, sucrose, and amino acids that are produced in the light phase. And another interesting result is that citrate is also produced in the dark conditions and fed to the light conditions as expected, and it happens in other models. And we can see here there is a flow amino acids and sucrose between the leaf and the phylogen through the inner bark, as expected by the common pool. And these results are really interesting because we were able to simulate the whole system, at least for now, with 3 tissues, and everything went as expected. So the future perspectives are to validate the secondary metabolism pathways that we have in this model, because we have a very rich secondary metabolism, it seems, when comparing with other models. We want to see if we can include other tissue-specific models to enrich our Diel model, and we are thinking on trying to see if we can use this model to analyze utilization of different nitrogen and phosphorus sources, to perform dynamic simulations with the model, because these simulations were all stoichiometric, performed with stoichiometric models for exponence analysis, and to eventually study the interactions with models from pathogenic organisms. I want to thank our team at Biosystems, namely these people that directly worked in developing the model, and also the team from ITQB, which it's not here, but we now are going to hopefully interact with them to help to further validate the model and to eventually include other data in this model. And thank you for your attention.
Thank you very much, Oscar. I now seize the opportunity to ask the audience if there are any questions regarding this topic. You can put it on the chat as well.
Can I make a question?
Sure, Nish.
Hi, Oscar. Thank you for your presentation. I'm just wondering if you can validate the biomass production that you show before.
Okay.
In this model, so the different proportions of different molecules.
Okay, thank you, Ines. So that allows me to clarify a part of the talk that was probably not that it didn't went that well. So in these cases, for microorganisms, we usually need to have really specific quantified biomass. We really need to know the specific percentage of each macromolecule and precursor, because we are calculating growth rates. In these models, usually we don't calculate growth rate because supposedly the purpose of these models is not to replicate biomass. So it has to replicate biomass to grow, but it's not the purpose as supposedly is in microorganisms. But we want of course, we want to have the biomass composition. We want it to be as precise as it can be. So we don't have, at least we didn't find, at least to the best of our knowledge, we didn't find the composition in terms of what we need, of the grams of protein per gram of leaf, for instance, for Cork Oak. You probably have that data, but we couldn't find it yet. But it's easy to swap to update the model. So that's the good part of it, is that it's easy to go there and update the biomass composition. And when I say to go the leaf biomass composition, any other tissue biomass composition is the same. So it's 1 or 2 reactions at most, and we just have to update the stoichiometry of that reaction. But when performing these simulations with these models for plants, usually the simulations are not for maximizing the growth rate. Usually the simulations are for minimizing the photon uptake, because we usually set a specific, we fix the biomass flux to one specific value. So we make the model has to produce biomass, but it produces at a specific amount that we set it to. So usually the amount is set to 0.1 because it isn't that important. But then the objective of the optimization problem is to minimize the photon uptake, because the premise that we have when we perform these simulations is that the cells evolve to use as less resources as possible. So when it's minimizing photon uptake, we are trying to guarantee that it will produce the minimum amount of energy that it needs to produce a specific amount of biomass and other products like oxygen in the case of them. But of course, that we would rather have better data that for the biomass composition for all the tissues. So for the leaf, we couldn't find any data specifically for Cork Oak, but for the phylogen and for the inner bark, we found some papers describing at least the amount of carbohydrates and sugaring and I don't recall which was the other one. And we knew that about 70 or 60 or 70% of the biomass was composed, for instance, in the case of the phylogen, 40% was sugaring and 40% was carbohydrates. And then we divided the rest by the same proportion that we knew that usually the plants are composed of, according to the other models in the previous one. I don't know if I answered the question. Hopefully I did.
Yeah, but in the end, you didn't show the suberin reduction. Yeah, I'm not sure you showed the citrate and rate, but you don't have any.
Yeah, because it's included in the biomass. It's not as a byproduct probably, but it is probably not included in. So the components of the biomass are usually used to create in the simulation a pseudo metabolite, which is called biomass. We didn't represent it on this schema, but I'm not sure, but I can check it in the simulations. We also are producing sugaring like a byproduct. Probably we are, but it's not there. But yeah, we can talk about that later.
Yes. Yeah. Thank you, Oscar.
Okay, no problem.
I think we have another question from Daniel Sobral. Do you want to put it, Daniel, or do you want me to read it?
It's fine. It's just a curiosity whether you really need to have a separate model for day and night.
Yes, because when we are simulating for night, we are not providing photons to the model. So since we don't provide photons, the metabolism will take other ways. So it won't produce oxygen, it will consume it. It won't produce carbohydrates, it will consume them. It will activate the glycolysis, not the gluconeogenesis. So the flux, the pathway distribution is really different. So in this case, we have the same model with different environmental conditions. That's why we consider it a different model, because there's that limitation. So that's one of the first tests that we make when validating this model. The Leaf model, for instance, is that we have the model, we provide it with photons, perform the simulation, and see if it uses the photons and produces oxygen. And if that's the case, then we can perform another simulation where we remove the photons and see if the model is also viable, but this time without the uptake of the photons, if it is also able to perform a valid simulation, though using other pathways, because it won't use the photons to produce energy, it will have to use the carbohydrates to produce energy and the other constituents. So in this case, in the end, the flux distribution, the pathways that are active are really different, and these are considered two different models.
Okay, thank you, Oscar. So we'll now move on and pass the word to Pedro Monteiro, which will talk about the YEASTRACT Plus portal. Pedro, please go on.
Hi. So I'll start by sharing my screen. Okay. So I will give a more historical perspective, and at the end I will show a bit of the recent developments of YEASTRACT. So YEASTRACT was based, or we started to study the Saccharomyces cerevisiae, since it's a model organism and it's quite well studied. It's the simplest eukaryote, and until 2005, our collaborators, the microbiology group at IST, had to go to different databases to perform or retrieve different data or do different functionalities. And this cost a bit of their time. It was a heavy load. And so they talked with us, and we started to collaborate and build a database and try to extract more information out of it. So the main message is that all these databases provide a very descriptive view. So if you go to SGD, you get some data characterizing a given gene. If you go to RSAT, you see data according to sequences, etc. And what we wanted was to explore a bit more about the regulatory information. And so given a given transcription factor, what we wanted is to check what are their target genes, and vice versa. Given a target gene, how is it regulated? And we want to visualize the network, if it's an activation, inhibition, and if there are many transcription factors involved, etc. And so this called for a mechanistic view of the data. And so we started to build a repository of regulatory associations for Saccharomyces cerevisiae. And so this has been going on for 15 years, more or less. And so we started developing in 2005 and curating many papers. Now we have more than 1,000 papers, which means that people at the Biological Science Research Group at IST have to read it and provide all the information, and for us at the NSKD side to load the data in the server and develop functionalities over that data. So the first release of YEASTRACT was in 2006, 1 year later, and then over the course of the years, we did a few updates where we always enriched the data with for the new papers that were coming out regarding regulatory associations in cerevisiae, and also developing new functionalities to exploit that data. So in 2008, we had the second release, and this is a photo that I retrieved in the computer from 2009, where we can check the targets of some transcription factors and visualize the network. So nowadays it's not like this, but it was just going back to in time. So throughout the years, we've got some recognition from the scientific community working in transcription regulation in cerevisiae, since we got to see accesses from the whole planet, basically. And so in the late 2000s, there were a lot of accesses. Then people start to shift for other model organisms, I suspect for human. And now we are getting an increase, but maybe because now we have more species integrated. So this also led to different joint publications, also supervision and training of students, and also research projects from the FCT, notably the BioData.pt. So regarding this descriptive view, so if we go to YEASTRACT, we can search for a given gene name or ORF name or some given keyword that is present on the description of a gene. And this is what we see. So we get the locus info where we have orth name, gene name, the ELISAs, what is the strain, the description, the genomic sequence, promoter sequence, and the coordinates in the chromosome. We can also see the information regarding the protein, the gene ontology, and so this is the information that we obtain from our source database. So in the case of Cerevisia, we are using YEAST-Genome, so the Saccharomyces genome database, so yeastgenome.org. And so for each yeast gene in the database, we always have the URL to go back to the source. So if we are in another species, of course, we'll go to a different database, but this way all the data is linked to the original source. So what we did was to enrich this descriptive view with a mechanistic view of the data. So we started to enrich with the regulatory association data. So every time we have a transcription factor regulating a target gene, we might have several papers supporting that regulation. And each of these papers will have an evidence code, which we grouped as direct or indirect regarding the experiment that was performed to acquire that data. So in the direct, we know that there's a direct binding of the transcription factor to the promoter of the target gene, and indirect, it's normally high throughput data that will not give us a direct evidence, but will give us a sign of the interaction. So the association type, if it's positive or negative. And that is also complementary to the direct information. And we also have the environmental conditions. So considering all the data that we gathered, we can now proceed to network visualization. And here we experimented a few things, and we settled with the JavaScript visualization framework, which is called D3. So people more computational should know it. And the code that we see here is, if it's dashed, it means that there's no direct evidence. And there's only supporting papers saying that is a positive interaction, but it's indirect. If it's dark black, if it's black solid, it means that there's a direct interaction, but no indirect evidence. And if it's solid red, means that there is at least one direct evidence and at least one indirect positive evidence. So you see that we start to combine all these informations to get a visual and easy to read representation. And so here the blue ones are transcription factors and the red ones are target genes. So with this, at this time we did the fourth release of YEASTRACT, where we have more data and better visualization functionalities. But then the need to explore other species started to arise, and in 2017 we did a release of YEASTRACT. It is a database containing 4 pathogenetic yeasts, so Candida glabrata, Tropicalis, Parapsilosis, and albicans, which account for 90% of candidiasis. And so by that time, we had to endure a major reshuffling of the database, because now we had to support multiple species and try to extract information by combining the information of every single species. And I will get to that in a moment. And so by that time, we started to have funding from BioData. And so we did the 5th release of YEASTRACT, but at this time we had 2 separate databases. So YEASTRACT, we had the 4 candidates, and YEASTRACT, it was like an academic database that was growing throughout the years with the problems that we all know that normally happen. So, which needed a major code refactoring. And so, with the support from BioData, we then, in 2000, so this year, mainly the work from last year, we did the YEASTRACT Plus release where we integrated everything. So the path of YEASTRACT, the YEASTRACT, and also non-conventional yeasts also were integrated. And we did a release where we have now 10 species. So all the species, so YEASTRACT, the classical for Cerevisiae, path of YEASTRACT for the pathogenic yeasts, and non-conventional yeasts, which have biotechnology interest. And so here the main idea is that now we have everything in the same place, and we can do we can link the homologous genes between them and extract information or the network for the most studied yeast for Cerevisiae, and try to infer some networks on the less studied yeasts, for example. So more concretely, imagine that we have two species here, and we can see this as a multi-layer network in graph theory, where we have a regulatory network in the space of species A. And then if we have another regulatory network for species B, we can now try to map the vertices of each network that will correspond to the genes, and do some plots and check the homology between all the vertices. And so what we did is that we have an in-house homology criteria where we ran BLAST-P, and then we say, well, every result which does not pass the 10 to the minus 5 p-value threshold will be discarded, which is not very strict, by the way. So 10 to the minus 5 is not strict, but we enforce that needs to be bidirectional. And so this one will clean a lot of false positives from the p-value. Because you see, if we go to a database and we check for the homologies, we might be in, for example, in the Candida genome database, we might be in a gene, check the homologous gene in the database, and says that it's this one in species B in another Candida, and then on the page of this gene, it will point to another, meaning it gives you the best hit for the best p-value, but it does not check for bidirectionality. And so here we chose to have bidirectionality. And also, instead of just accounting for the best hit, we have a tolerance of the BLAST score of 10%, where we also include blasts that are less than 10% away of the best BLAST score. And so this way, we can account for gene duplications and check homologous that are that are closely related. We then have an additional level where we account for the synteny also of the genes, and the user can choose if he wants homology only based on BLAST, or also additionally on synteny. And so it has to have 1, 2, or 3 neighbors at each side in a window that is, I think, if I recall, 15 for each side. So with this, now we can do cross-species network comparison, which, for example, we can see if we focus in a given process. So we have a set of at least one transcription factor or a set of transcription factors and their target genes. according to some environmental condition. And then we can check the same network in another species and then compare it. And so, and easily select the network only in one species, only on another species, unique to a given species, unique to the second species, or common to both species, and see the common regulatory network between them. Also, we can filter by supporting evidence or type, of course. Another interesting feature is that sometimes we don't have the network, or as we had more species into the database, the new species don't have any regulatory association added. So they are species that are not very well studied in the literature, and we don't have any supporting evidence. And so in this case, we do network inference. So we project the network from the most studied species into the vertices of the less known species. So imagine that in species B, we don't know the network. And so we can project the network for Cerevisiae, for example, and project it on the species B. And like this, we can infer what is the potential network in this new species according to, so sometimes you don't have homology to map the vertices between the species, but for those that you can map, you can project the network also. And so this came in the release of 2020. So now the question that we can pose is, so what happens if we want to analyze a newly sequenced genome? And so since we had the experience of refactoring the whole database and integrating more species and particularly more strains also within a given, the same species, we started to look into it and try to make so to automate the whole procedure of loading the new genome. And so we did this for rhodothorax uroides that is non-pathogenic and has a biotechnology interest, and we have the complete genome assemblies in GenBank. And there was no previous database or tools available specifically for this species. And so what we actually did was to strip YEASTRACT from all the particularities and to provide all the source code and database schema open source, so you can check it and download it, and provide all the scripts that can process a genome assembly from GenBank in the GBFF format. So with this, then we can provide we also provide the scripts to compute all the homologs against the reference genome. So we can imagine that someone from across the globe can use this for some other species, for example. If he has a reference genome for a given bacteria and wants to study a newly sequenced bacteria, he can do or can profit from the whole infrastructure. We also provide the scripts that can compute the TF binding sites from the reference genome that we already know, so that the given transcription factor has some associated binding sites. So if that information is there, we immediately compute the matches against the promoters of the newly sequenced species to try to also infer potential regulatory element conservation in the promoter, and also the functionality to infer and visualize networks. And so we did a case study for this and we submitted for publication recently, and hopefully it will come out next year. So this is an example of rhodothorax uroides where we have a given transcription factor, in this case HAA1, where we here see the network in Cerevisia, and we immediately see that we have two transcription factors. So there are two copies of the gene in Cerevisia, one in Zebaili and one in Glabrata. And we can see in the network itself, in the visualization, what is the name of the genes in the target species and in the original ones where we are. So if we are in Rhodotorula or some other newly sequenced species, we can see immediately a one-to-one mapping to when we are working with the network. And also we can interact with the visualization, like I shown previously, where we can hide the activations, the inhibitions, or visualize only the DNA binding or expression, etc. And so with this, we have like an official release of what we call community ELIXIR, and it's separated from the other ecosystem, so the rest of the portal, because in here, these three databases, the non-conventional, the pathogenic, and the classical ELIXIR, we do provide curation for the regulatory associations. So in community ELIXIR, we can insert a genome, but we don't provide curation. So we maintain the infrastructure, but we don't populate the database. And so hopefully into 2021, we'll have the official release of community ELIXIR. And so with this, I call for the community, if you want your genome also available in the portal, you can submit it. So you can ask us to put it, or you can host it yourself on your own web server, okay? So we provide the two versions. And I want to thank everyone involved since 2005 on the YEASTRACT and their sister database, because so it was a joint effort, so present and past contributors and all the funding that we have. So thank you.
Okay, thank you very much, Pedro. Are there any questions regarding YEASTRACT? You can also put it on the chat if you want to.
Another curiosity of mine, if you don't mind.
Sure.
How do you position yourself regarding, for example, ensemble fungi and things like that? It's more a political thing, but I guess.
In that respect, I don't know. So I cannot answer fully because I don't know exactly all the information that they provide. But for example, for East Genome, a few years ago, they started to provide regulatory information. And so we thought, well, maybe, so they are the reference database, they will provide everything, and we'll close YEASTRACT. But actually, so we have a good interaction. We provide them with information, and they provide us with information, and it's actually complementary. So, but for ensemble, I cannot answer fully. Maybe Miguel can answer.
So we don't try to compete with them because we don't have the same purpose. I mean, we will never extend YEASTRACT to all fungal genomes, then we don't want to do that. And they don't explore the kind of data that we explore, which is focused on transcriptional regulation. So, and of course, I guess they have an enormous budget to keep the database, and we're doing it almost as a hobby, no longer as a hobby since BioData, but so we're keeping focused on a specific aspect of the biology of these organisms. We are extending progressively to other systems, but we don't intend to compete with them for a similar database.
Okay, I guess there are no more questions. Okay, so thank you, Pedro. And I would also like to thank Miguel Rocha, Miguel Teixeira, Isabel Sá Correia, and Isabel Rocha for embracing this challenge. And let me just share. Pedro, can you stop share your screen, please?
Yeah.
Thank you. Okay. Sure you can see my screen now. Okay, so I would like to announce the next BioData talks, which will be about the BioData Marine Metagenomics Community. And we will count with the presence of Simon Cox and João Machado from CCMAR. And I would also like for you to, if you want to register our forum and subscribe our newsletter at the www.biodata.pt. And thank you all for your presence and hope to see you in a month. Thank you.
Bye.
Bye, everyone.
Bye.
Bye. Thank you.